So let us move to, if you have any question, just let me know. Okay, I'm happy to answer the question. So now we move to the chapter 11, transmembrane transport of ion and the small molecules. So again, those molecules need to transport, it all happen to the plasma membrane. Now we previously just kind of introduced how molecules stably incorporate in the membrane located here. But now we want to know how molecule transport across this hydrophobic core. We first just focus on the pure phospholipid bilayer. We just check their chemical or physical property first. So those lipids, they can form the beautiful phospholipid, but they still have some space. Okay. So which component can easily penetrate this lipid bilayer would be the small one. Super small one, like a gas, CO2, N2, or O2, oxygen, can easily go across the membrane. The membrane cannot stop them, okay? Because it's a hydrophobic, the chemical is a hydrophobic, so the charge and the hydrophilic molecule cannot penetrate this hydrophobic core, okay? So the pure phospholipid bilayer, they are impermeable to the large charge and the hydrophilic molecules, okay? So gas, because they are small, they are permeable to those phospholipids. No one can stop them. The small uncharged polar molecule ethanol is permeable, okay? And the water, urea, they are slightly permeable, okay? But the large uncharged one, glucose and the fructose, they are impermeable. Or ion, because they are charged, they are impermeable. Or the charged polar molecule, amino acid, ATP, glucose, protein, nucleic acid, they are all large molecule. They cannot kind of really permeable for those pure phospholipids. And just remind you, the passive diffusion. So this is the one tank separated by the two areas, and you will be this molecule always will transport from the high concentration to area with a high concentration to the low concentration until they reach the even distribution or same identical concentration. We call this. They reach dynamic equilibrium, okay? That's what be driven by the passive diffusion. So it also happens here. What's the factor to determine their transport rate or transport speed? If the concentration of a certain molecule, the concentration between these two sides difference is large, they will be kind of transport faster. We call that concentration gradient, okay, here. Concentration gradient. Of course, the transport factor also determined by whether their hydrophobicity or the size. More hydrophobic one, more smaller one will be easier to penetrate the plasma membrane. So if you design, that is why people tend to kind of, when they design a drug, they try to design a small molecule drug because it will be easier for cell to absorb the drug, okay? Otherwise, they cannot go into the cell. Nothing happened. Okay, yeah. So you need to design a small one. So hydrophobicity and the size of the molecule determine their whether permeable rate. And the concentration gradient depends on passive diffusion. It will also determine their transport across the bilayer. Of course, and not only concentration, their charge also determine such a transport way. If there are many positive charge here, negative charge here, they will tend to kind of reach the balance. So electrostatic situation status will also determine. So we usually need to consider both electro and the chemicals. So that is why we usually consider the electrochemical gradient together, okay? So electrochemical gradient will determine whether molecule can cross the membrane, whether the speed is go fast or slow. So based on the passive diffusion, this principle, after a while, the process, if the cell only use a pure phospholipid bilayer, they will reach, at certain point, they will reach dynamic equilibrium. Okay, that means the permeable molecule, they will have a same concentration inside and outside the cell. And nothing happens. We cannot generate specific reaction inside the cell. There will be no living thing. We don't have any different outside the environment, right? So it's not the case. So that is why we need some protein to help cell to generate specific uptake and release specific molecule under the control those will be this protein. Okay? So ATP power pump, ion channel, and the transporter. So based on this cartoon, you need to understand this will be the gray will be the membrane, and this triangle, pink triangle, will be to show the electrochemical gradient of the pink molecule. This will be higher electrochemical gradient of the pink one, and lower electrochemical concentration in this side. Okay, so based on passive diffusion, this molecule, if they transport from this side to the other side, they don't need the energy. They're only driven by the passive diffusion. But if they want to transport from this side to the other side against electrochemical gradient, they always need energy. There is no exception because this is a universal principle. Okay, so there will be kind of several definitions for the ATP power pump. So they will use ATP hydrolysis. So ATP will hydrolyze to the ADP and the inorganic phosphate group and use this energy to pump the pink molecule against this electrochemical gradient to the other side. They need energy. For the ion channel, they don't need energy. They only kind of control whether it's open or closed. Open or closed, okay, yeah. So it just provides an easy way, shortcut for the molecule. But the transport motivation, the driving force, still follow its electrochemical gradient. Okay, so they don't need the energy. Okay, and for the transporter, there will be unipolar or sympolar or antipolar. For the unipolar, it's only transport one molecule. So, but unipolar only transport follow its electrochemical gradient. It's called unipolar. That it doesn't need the energy. But the sympolar and the antipolar, they will co-transport at least two molecules. So the sympolar, they will transport two different kind of molecules in the same direction. And the polar will transport two molecules with opposite direction. And this sympolar, they will transport here. They will transport the pink molecule, follow its electrochemical gradient, and use this as energy to transport black one against its electrochemical gradient. Okay? And then the polar, again, will use to transport the pink one, follow its electrochemical gradient, and use this energy to transport the black one against its electrochemical gradients. Okay, so this doesn't need energy. This doesn't need energy. These two and this one need energy, but they use a different energy source. Okay, so with the one need energy, we call the active transport. For example, this will be the kind of active transport. And these two do not need energy, but they still can kind of provide shortcut. They still can help, okay? But they are not the driving force. They, whether the molecule can transport or not, it still depends on their electrochemical gradient. We call this a facilitated transport, okay? And for this sympolar and the antipolar, they still active transport, but just try to make a different with those ATP driving cases. So we call the secondary active transport. Okay, they use energy from the one molecule they transport, not the ATP hydrolysis. Okay, so yep.
Last time. So the B and C having the same function.
They what?
They are doing the same thing.
Yes, just transport to a different direction. So in this one, sympolar, they transport in the same direction.
So that's why the name is changed.
Yes, yes, yes. But again, their goal is try to transport their target. For example, here will be the black one against the electrochemical gradient. Yeah, okay. Unless if you see anything kind of against their electrochemical gradient, they need energy. Okay, there's no exception. Okay. Okay, so many kind of such a membrane physical or membrane chemical or electrochemical gradient would be kind of follow in the entire membrane. So the membrane protein, unless they in the same pool, they will functional together. For example, this will be a sodium-potassium pump, which is ATP power pump. They will use ATP hydrolysis to pump more sodium and generate sodium electrochemical gradient of the sodium here. And pump more potassium here and also generate the electrochemical gradient of the potassium. So in this other ion channel or sympolar can follow this electrochemical gradient to release the potassium from this side to the other side. okay? And the symphoter will use this sodium electrochemical gradient of the sodium to transport sodium from this side to the other side, and use this as energy to transport lysine from this side to the other side, okay? So one guy's generate electrochemical gradient can kind of affect others, can driven the reaction of others, symphoter or ion channel. Okay, so this is just table give you the definition. First, simple diffusion doesn't need any specific protein, they just through kind of physical or chemical principle. Facilitated transport, active transport, and the co-transport or secondary active transport, they need a protein we introduced before. And if this only the active transport or co-transport, they again is a gradient. And ATP active transport, they use ATP hydrolysis as energy, and this a secondary active transport they use kind of driven by the movement of the co-transported ion down is a gradient. Okay, they use a different energy source. Okay, so we just really quickly to go through what's the physical or chemical property of the pure phospholipid bilayer. So larger and hydrophilic molecules are difficult to diffuse across the membrane. Okay, so we need a protein to help us to generate, transport specific molecules to their destination. So several pump, ATP power pump, on channel transporter can transport this specific molecule across the membrane. Okay. So first we focus on the unipolar. So we introduced that by compare the unipolar with a simple diffusion. Simple diffusion is just pure phospholipid, no protein there. What's different if the phospholipid have unipolar or no unipolar? Without the unipolar, the diffusion rate will be the slow. The unipolar transporter will be the fast. They kind of provide a shortcut, help blend across the membrane. And it depends on the number of the unipolar. If on the membrane there are more unipolar, they kind of can provide more shortcut. So theoretically, they kind of can transport the ion or the cargo from the one side to the other side more quickly, okay? And the specificity, there's no specificity here. It totally depends on, for the simple diffusion, it totally depends on hydrophobicity and the charge and the size, okay? But the unipolar transport only allowed specific cargo to go across their kind of protein, okay? So it's specific. And direction is both reversible. What does this mean, reversible? It depends on electrochemical gradient. It can go this side and it can go the other side. It totally depends on the electrochemical gradient, okay? Once this change to the other kind of triangle become this triangle, it can also go to the other side, okay? So this would be the kind of one example for the unipolar is a GLUT1. So GLUT1 is the unipolar to transport glucose, okay? So this will be the ketone of the GLUT1, and this will be the electrochemical gradient of the glucose. There will be higher glucose here and lower glucose here, and there will be the one glucose binding site here. Okay, so first you need to know this binding site. You need to know those binding. The glucose will not continuously bind there and then don't go away anymore. No, there's no such a case. It's all about affinity. If you have it's always kind of cargo or substrate always bind that and deassociate. Bind there and deassociate. It depends on the affinity. It will be K of, right? K of rate, something like that. It will bind and deassociate. If you have a stronger affinity, it will retain on the binding side more and less deassociation. If you have weaker affinity, we have a more deassociation but less binding. Okay, it's a kind of dynamic situation. So again, it's also happened here. It will keep binding and deassociate, and also this GLUT1, as a member protein, the GLUT1, they don't need energy, but our body is a warm environment and they can kind of generate thermal motion. So this kind of protein conformation will kind of keep changing their conformation like this. The energy comes from the environment. It's not come from any other thing directly donate to this protein. Okay, so this protein will, if you keep your body temperature to the 37 Celsius degree, they will keep doing this. Okay, they will keep changing this conformation and this conformation. And so they will open to this end and the other end because those binding side, the glucose will bind and deassociate, and gradually they will move to the other side. And it's still the direction, transport direction is totally just follow the glucose, electrochemical concentration of this glucose, okay? It just provide the binding side and keep open to the other side or the other side, like this, okay? That's the way unipolar to transport molecule, and it's specific because that binding site only designed for the glucose. And we also need to kind of introduce really quickly about the osmosis, kind of osmotic pressure. It happens with a certain kind of defined factor. First, you need to have water. And second, you need to have, in the system, you need to have a selective permeable barrier, okay? So this is a U shape. You kind of have learned that when you was young, okay? And this, you will see the blue color is the water molecule, and the green one will be the subsolute, which is a sugar. And the water like to bind sugar, okay? And there will be kind of, in the middle, there will be a kind of selective permeable barrier. It's only allowed water to go across, but sugar cannot, okay? And in the case, if you put more solute, more sugar here, and less sugar here, sugar cannot cross this barrier, so there's no such a passive diffusion. But the only way to kind of, again, to solve this issue is more water, because water can cross, more water will go here and bind to more concentration of the sugar. That is why you have a higher water level here. And such a difference here would be the osmotic pressure for you to visualize osmotic pressure, okay? So it's only happened to the water can go across certain selective permeable diffusion barrier, and then there will be in the two different sides, there will be the solute different, okay? More solute allow the water to bind there, so the water will go out, okay? So that it also happens to the cell, okay? If you have a higher concentration of the solute outside the cell, the water will go out. We call this hypertonic solution, okay? And the cell becomes shrink. If you have a higher solute which can bind with the water inside the cell, the water will go in, okay? And they keep absorb the more water, then finally it's sweat out and break down, okay? So we call that hypotonic solution. If you have the same concentration of the solute inside and outside of the cell, it will be the isotonic. This event we call it a tonicity, okay? Hypertonic, and isotonic, okay? But water only slightly permeable to the pure phospholipid barrier. Who can help? This only happens when water can easily go across the membrane. So who will be the guy to help this? Aquaporin that I introduced before, okay? So this will be the aquaporin. This is the top view of the aquaporin. This is the one monomer of the aquaporin. And they always form the tetramer with the 4 aquaporin assemble together and do their job together. Of course, they are transmembrane proteins, so you will see a lot of alpha helix here, okay? And the surface is hydrophobic, as I mentioned before. But the interior here will be the water tunnel here, okay? So, from the section, you will see there's a tunnel here. So the water, and the diameter is about 0.28 nanometers. So it's perfect for the water molecule to go across here, okay? But they still have some charge amino acid here. So if a certain smaller ion, for example, the proton, want to cross this area, they will be kind of bind with those charge amino, and they cannot really go across this ion chain. It's only good for the water molecule, okay? Yeah. So, so this will be the cartoon to show you the water will gradually go through these tunnels, and the movement of the speed of those water movement and the direction is totally depends on osmotic pressure, whether they are more solute for water to bind or whether they are more solute to bind here. Okay, it totally depends on that, because the solute cannot cross this. Water will be the only one can move. Okay. Okay, so this part we introduce the unipolar, is faster compared to the simple diffusion. It's reversible. It totally depends on it, follow its electrochemical gradients. It's specific. It's only transport-specific molecules because they have a specific binding site. Two examples of the unipolar. One is GLUT1, so it will keep changing their conformation to transport glucose down its concentration gradient. Aquaporin will provide a channel allowing water molecule to move across the membrane. The diffusion direction depends on osmotic pressure. Okay? Yeah. Any questions so far? Okay, now we move to the ATP power pump, which use ATP hydrolyzed as energy to deliver molecules. So the ATP will be the kind of energy carrier. You put the 3 phosphate group here. The phosphate group have a strong negative charge. If you want to put the 3 negative charge guys together, you can imagine there will be really strong bond required to connect them, right? So if you break down this bond, it can locally release the energy, okay? So that's the way kind of cell will use this to synthesize many ATP as they to deliver their energy. When they across to the certain enzyme or certain protein, they can break down this bond and locally release the energy for them to use. And then to, for example, the ATP power pump can transport this pink one against the electrochemical gradient. There are several ATP power pump transporter, transport protein, for example, P class, V class, F class proton pump, and the ABC superfamily. So they are the guide to transport or generate certain electrochemical gradient, and the other protein can follow this to do their job. So you will see if they check, this is invertebrate cell, this is the vertebrate cell, what's the kind of ion concentration inside the cell or outside the cell, which with the blood here, is quite different, okay? That will be contribute mainly from ATP power pump. So you need energy to generate certain specific environment to do those living things. Okay, so let me show you these videos. It's from the textbook. They claim that releasing the calcium from the sarcoplasmic reticulum can induce muscle contraction. Just watch this video.
Muscles every day to do activities. This woman is using muscles to breathe, circulate blood, and move her hand to take notes. Your cardiac and smooth muscle tissues are involuntary. You do not consciously control their actions. Skeletal muscle works under voluntary control. Skeletal muscles are composed of bundles of muscle fibers. Muscle fibers are long cylindrical cells containing several nuclei. Muscles will contract or relax when they receive signals from the nervous system. A neuromuscular junction is the site of the signal exchange. This is where the synaptic bulb of an axon terminal and muscle fiber connect. Muscle fibers are composed of many myofibrils. A myofibril contains contractile units called sarcomeres. Sarcomeres run adjacent to one another down the length of the myofibril. Each sarcomere consists of alternating thick and thin protein filaments, giving skeletal muscle its striated appearance. The muscle contracts when these filaments slide past each other. The thick filaments are myosin, which are anchored at the center of the sarcomere, called the M line. The thin filaments are composed of the protein actin, which are anchored to the Z lines on the outer edges of the sarcomere. Because the actin filaments are anchored to the Z lines, the sarcomere shortens from both sides when actin filaments slide along the myosin filaments. Although the action between the filaments is described as sliding, the myosin filament actually pulls the actin along its length. The cross bridges of the myosin filaments attach to the actin filaments and exert force on them to move. This action is known as the sliding filament mechanism of muscle contraction. In this model, the sarcomeres shorten without the thick or thin filaments changing in length. A contraction begins when a bound ATP is hydrolyzed to ADP and inorganic phosphate. This causes the myosin head to extend and can attach to a binding site on actin, forming a cross bridge. An action called the power stroke is triggered, allowing myosin to pull the actin filament toward the M line, thereby shortening the sarcomere. ADP and inorganic phosphate are released during the power stroke. The myosin remains attached to actin until a new molecule of ATP binds. freeing the myosin to either go through another cycle of binding and more contraction, or remain unattached to allow the muscle to relax. Muscle contractions are controlled by the actions of calcium. The thin actin filaments are associated with regulatory proteins called troponin and tropomyosin. When a muscle is relaxed, tropomyosin blocks the cross-bridge binding sites on actin. When calcium ion levels are high enough and ATP is present, calcium ions bind to the troponin, which displaces tropomyosin, exposing the myosin binding sites on actin. This allows myosin to attach to a binding site on actin, forming a cross bridge. Calcium ions are stored in the sarcoplasmic reticulum and are released in response to signals from the nervous system to contract. Neurotransmitter molecules are released from a neuron and bind to receptors, which depolarizes the membrane of the muscle fiber. The electrical impulse travels down the T tubules and opens calcium stores. Calcium ions flow to the myofibrils, where they trigger a muscle contraction. As the actin and myosin slide along each other, the entire sarcomere shortens as the Z lines draw closer to the M line. As the sarcomeres in myofibrils contract, the entire muscle fiber will shorten. When muscle fibers contract in unison, a muscle can produce enough force to move the body, allowing you to take notes.
Okay, so just a reminder really quickly. So the neurotransmitter will depolarize the membrane and then will trigger the calcium stored in the sarcoplasmic to release the calcium, and the calcium will bind to the troponin. So it will change and release the myosin binding site on the actin filament. So the myosin will bind to those actin filament and trigger the muscle contraction. That will be the mechanism how we can use our mind to do the muscle contraction. Okay, and the point is how can cell accumulate so many calcium in the sarcoplasmic reticulum is through the ATP power pump. Okay, so this will be the sarcoplasmic reticulum lumens. As you can see here, there will be higher electrochemical concentration of the calcium here compared to the cytosol. And also this will be driven by so-called P-class pump. So the P-class pump is kind of one family of the ATP power pump. They share the really similar kind of protein structure. They will have an ATP binding site here, and the one amino acid need to be phosphorylated to change their conformation. There will be two different conformation, E1 and E2. Okay, for the E1, of course, there will be the one sodium binding site here, and they have a strong affinity with the calcium binding. Okay, with this, and then the ATP will bind here and do the hydrolysis and release ADP, and it will put the inorganic phosphate group to this site. Okay, to phosphorylate as aspartate here. So why they call the P-class pump? Because all the member here, they need to be phosphorylated in that site. So that is why they call P-class pump, okay? And then once they be phosphorylated here, they will change to the E2 site, E2 conformation. And the E2 conformation, the tunnel will face to the other side, and their affinity will become weaker with the calcium. That is why they will release the calcium. Okay, so they will change to the E1 and with calcium bind and change to E2 and release the calcium. That is why they gradually can use ATP hydrolysis as energy to change the conformation and keep pumping the calcium to the SR lumens. Okay, and this is a cartoon of the sodium transporter, ATP power pump, ATPS. You will see here, will be the binding site, and this will be phosphorylation site, and ATP bind here, and it will go to the other. Okay. Then you can find this calcium ATPS to the different subcellular region. For example, it's on the plasma membrane, keep pumping, use energy to pump out the calcium to the extracellular space. It will also pump a lot of calcium from cytosol to the ER lumen or in the mitochondria matrix. Okay, so in the end, the concentration of the calcium in the cytosol will be super low. That is why, if you check here, they always have a big difference of the calcium in the inside the cell compared to the outside cell. Will be maybe 1,000 or a couple thousand fold different. Okay, so that is why when the calcium go into the cytosol a little bit, cell can sense such a difference. and then trigger downstream signaling. That is why the calcium can functional as a secondary messenger. It's not the ligand. It just triggers many calcium-dependent signaling. Because it's a little bit calcium going in there, they can sense the difference. Okay. As I mentioned before, the P-class pump, they share the pretty similar protein structure. For example, the golden one will be the 3D structure of the sodium, potassium, ATPase. The purple one will be the calcium, ATPase. They almost identical, just a little bit different. Okay, so that is why with the cartoon, you will also still see the 3 balls here and the one column here. Okay, and this will be the sodium, potassium, ATPase. So it both contain, again, it contains 2 different conformations, and the E1 will allow the sodium to bind here, and E2 they will release the sodium, and they also have E2 have a strong potassium here, but have a weak potassium here. That is why they will transport potassium here and sodium to the other side here. Okay, and such a conformation change also driven by the ATP hydrolysis and the phosphorylation of this side. That is why they all call the P-class pump. Okay, and next, this will be the V-class proton pump. This pump only transport proton. And why it's called the V-class proton pump? Because you always express in the vacuole or the vesicle structure like a lysosome, endosome, and the plane the vacuole. Okay, their function is mainly acidify the lumen, make it more acid. Okay, of those organelles. So for example, this is could be the lysosome maybe. They try to acidify this lumen. What's the definition of the pH? The concentration of the proton, right? The higher concentration of the proton will be lower pH level. Okay, so if you use ATP hydrolysis as the energy to transport more proton here, you will accumulate more proton, then it will lower the pH level. Okay, but it's weird. They show this. You just keep transport the proton here, but it still keep the neutral pH. It doesn't acidify the lumen. It's only happen when you transport proton and both transport chloride here. They will become acidified, acidic proton pH. Why? Because if you the ATP-driven ATP power pump, they transport proton pretty slowly. Okay, they use energy and transport proton here, and it will generate. If they have a charge here, they will generate electrochemical gradient, and there are many unchanneled or unipolar can easily release the proton outside those vesicle if there's an electrochemical gradient here. So it will be easier to leak out. So you cannot accumulate enough amount of the proton here. Okay, and such a pumping is super slow. It's only happen if you transport chloride here, and we will conjugate become HCl. They will no more kind of charge there, and there will be no more electrochemical gradient here. Then those unchanneled unipolar will not take this function to leak out the proton. That is why you can make it more acid. Okay. Okay. So next will be the F-class proton pump. Let us finish this part, and we can kind of finish the course. Okay. So F-class proton pump, basically they are the guys to synthesize ATP, okay, to generate the energy. So you can find that in the mitochondria, which is the power factory to synthesize the generated energy. So this is the inner membrane of the mitochondria. You will see after glycolysis will collect many energy from the sugar, from the food. They will put the energy to those kind of electron carrier NADH. Then go into the mitochondria, and the NADH will transfer those high energy electron to different protein. We call it electron transport chain. They will use this as energy to keep transport proton to the intermembrane space. Okay, then generate a good kind of higher electrochemical gradient of the proton here. And the F-class proton pump here, or we call it ATP synthase, depends on based on its function. It will follow. There will be tunnel for the proton to go across, and use this electron transport electron chemical gradient as an energy to change the conformation, and finally can kind of conjugate ADP with the organic in phosphate group and become the ATP. That is the way they synthesize the ATP. Why they call the F-class proton pump? Because it really doesn't make sense. It's energy coupling factor in mitochondria, and they choose F factor to claim is a F class. Doesn't make sense, right? Okay. There'll be many, many other factors why they choose factor. Okay. So, but that's all kind of testable told us, right? But there will be one weird thing here. Okay. This is inner membrane here, and there will be a lot of electron transport channel keep pump proton to the intermembrane space. But here, the outer membrane of the mitochondria, there will be a lot of protein called porin, or a lot of transporter, a lot of pore there. And it will allow the molecule smaller than 5 kilodalton or smaller than 1.5 nanometer in diameter. They can easily across outer membrane. Okay, the proton is super small. They can easily go in and go out. They cannot really keep the high concentration or generate this electrochemical gradient of the proton in the intermembrane space. So there's no way for mitochondria or electron transport chain to generate such a electrochemical gradient. Okay, so how to kind of explain this? But mitochondria can really generate energy. Yeah, right. So the thing is, it only happens to the cristae lumens, okay? Because when there's a kind of standard answer, what's the function of the cristae? Do you remember? Why we need a cristae of the mitochondria? Because it can increase the surface area, right? It's not to me, it's half right, okay? Why we need the cristae? Because they can generate closed environment. If you pump to the intermembrane space, we need to separate the two area. Okay, this will be the cristae lumen here, and this outside the cristae will be the intermembrane space. If you electron transport chain pump the proton to this area, there will be no chance to generate electrochemical gradient. But if they pump to the cristae lumen, they can generate the electrochemical gradient because they can form the junction here. Okay, let me show you the result. There are many proteins will make this really tight and close this, make this become the closed environments. Okay, that is why if electron transport chain, they keep pumping proton here, can generate here. If they pump here, nothing happen. Okay, yeah. So if you check the electron kind of potential, because several dyes like a TMRE can measure the membrane potential of mitochondria. This is one mitochondria. You see this is a cristae, and this outer will be the intermembrane space, not the cristae lumen. You will see the warmer color means higher membrane potential, which means the higher proton electrochemical gradient. The cooler color means lower membrane potential. You don't see the strong membrane potential in the intermembrane space. You only see that strong membrane potential in the cristae lumen. Okay, and they are separate environment. They function independently. Some cristae have a strong, some cristae have a lower membrane potential, right? Depends on how many electron transport chain are located in that cristae membrane. Okay, so that is why you need to. The answer is you need to kind of put the electron transport chain to the cristae. That is why you need a cristae. If you put here, no, your mitochondria cannot generate energy. Okay, get the point? Okay. Okay, so this will be the cartoon to show the the ATP synthesis, how it can synthesize the ATP.
The flow of protons down their electrochemical gradient drives a rotor that lies in the membrane. It is thought that protons flow through an entry open to one side of the membrane and bind to rotor subunits. Only protonated subunits can then rotate into the membrane, away from the static channel assembly. Once the protonated subunits have completed an almost full circle and have returned to the static subunits, an exit channel allows them to leave to the other side of the membrane.
In this way, the energy stored in the proton gradient is converted into mechanical rotational energy. The rotational energy is transmitted via a shaft attached to the rotor that penetrates deep into the center of the characteristic lollipop head, the F1 ATPase, which catalyzes the formation of ATP.
Okay, so basically there will be the kind of binding site for the proton. So once the proton will go through this pathway, follow its electrochemical gradient, and try to rotate, and this force will rotate this kind of ATP synthesis, and it will kind of also rotate the gamma subunit after this, beyond this, below this transmembrane domains. And rotation of this gamma subunit will kind of change the ball like a structure alpha and beta subunits. So if you from the top view, this will be the gamma, okay, and this will be the alpha and the beta. There will be a 3 pair of the alpha and the beta, okay. So it will keep rotating and change their conformation, okay, and there will be 3 conformation kind of transition for those 3 pair. So this is 1 pair containing alpha and the beta, okay. This will be OLT conformation. O will be the open conformation, which is ready for, it's open to the ADP, and the organic phosphate group can freely go in and ATP can go out. It's open conformation. But once they change, the gamma rotate, they will change to the L conformation. So the ADP and the organic phosphate group will loosely bound here. They will retain there, okay? But once they change to the type conformation, which stands for the T, will make a ADP and the organic phosphate really close to each other. So chemically, they will spontaneous to form the ATP based on this. Then they will shift back to the open conformation, then release the ATP. That's the people suggest, okay, how such a conformation change can generate the ATP. So the 360 rotation will generate 3 ATP, okay? So how to prove they are really kind of rotate? Here, I just want to know this is really smart machine. If they generate many or enough ATP, they actually can also serve as an ATP power pump. They kind of ATP will help them kind of to change the conformation and the kind of rotate with the opposite direction and pump back the proton back to the crystal lumen. Okay, so if they don't have enough ATP, they will go this direction. If they have enough or too much ATP, they can pump back to the crystal lumens. So how to prove that? They just several researchers, they just purify the kind of alpha and the beta subunit and code them on the cover slide, okay? And put the gamma here and just add ATP. So then they will kind of rotate, right? But this tiny dot, if it rotates, you cannot see any difference. So they just conjugate a fluorescence labeled actin filament here, like a helicopter. You add ATP, you start to see such a kind of actin filament keep doing, rotate like a helicopter, okay? Very elegant experiment. Okay, so you will see this ATP or F-class proton pump. They can be found in the intermembrane or thylakoid membrane in the chloroplast. Here they will generate electron transport chain will transport to the intermembrane space, but I think it should change to crystal lumen. Okay, and thylakoid space there's no it's correct here. Okay, but for the mitochondria it should be the crystal lumens. Okay, and use this electrochemical gradient to generate, to rotate their conformation and then generate ATP. That would be kind of the way to synthesize the ATP. So we just quickly introduce what's the P-class pump. They use phosphorylation to pump the specific ion to the certain lumens. And also V-class, they will retain in the vacuole or the vesicle. They will kind of pump the proton to the lumen. You need to come with the chloride, otherwise it will be easier to leak out and it cannot acidify the lumens. And the F-class proton pump, it will locate in the mitochondria, follow the electrochemical gradient of the proton, and it can rotate such a structure and then generate, combine the ADP and the organic phosphate group to become the ATP. That is why it's also called the ATP synthesis. So we probably will stop here, and next week we will introduce ABC superfamily. Yeah, so that would be the kind of what I would like to share today.